this is the last of the categories of macronutrients, right? We've been covering the macronutrient. We started with elements in the body and how that relates to agriculture, and then why various elements are so important. That was a doorway into talking about the macronutrients. We started with DNA, which led into a discussion of GMOs. And then we talked about proteins, amino acids and proteins. The next category of macronutrients was carbohydrates. We just finished that. And then now the last category of macronutrients is lipids. When we're done with this, the topics I still like to cover would be calories and metabolism, changes to micronutrients, and changes that happen to food, cooked food. I think I can get those topics, at least a little bit of them, before the end of the semester. All right, so lipids, the general category of fats and oils, that kind of molecule is called lipids. They're lipids. And lipids are, they have a number of functions. However, they are primarily a form of energy storage, a form of energy storage and energy supply for muscle, liver, and importantly, the kidneys. All of these foods that you see here are examples of foods that are high in different kinds of fats or oils. So everything from bacon and mayonnaise with its egg in there and heavy cream, to olive oil and milk and the fats that are found in vegetable sources like coconut or avocado. All high in different kinds of fats and oils. To begin with, I'm just going to try to give you the language that you need to be able to talk about fatty acids or lipids in general. I'll show you the structure of a number of different categories of lipids that you've probably heard of. You've probably heard of fatty acids. You've probably heard of cis fats or trans fats at least. You've heard of saturated and unsaturated fats, maybe polyunsaturated fats. Maybe you've heard of triglycerides. But I'm going to help you understand what all of these words actually mean. So a fatty acid is the fundamental building block of, or fundamental category, I should say, of biological lipids. In general, a lipid is going to be some kind of what's called amphipathic molecule. Amphipathic, it just is a word. So like an amphibian, it's the same prefix. An amphibian is an animal that lives in the water and on the land, right? And an amphipathic molecule is simply a molecule that has polar and nonpolar regions. So there's going to be parts of the molecule that like to be in water and parts of the molecule that do not like to be in water. And we can take these fatty acids and build them into more complicated structures. We'll see that in a moment. For now, when we look at a fatty acid, there's going to be two regions to it. The first region, the one that gives it its sort of greasiness, is this long hydrocarbon chain. And we represent that just by this zigzag line here. At each of those junction points, at each of those joints, we have a carbon. So for example here, it would be, and we can see this zigzag line as you go along here. And then each of those carbons has a bunch of hydrogens attached to it, though, as you see here. We're not drawing them in, right? The only hydrogen that we're going to draw in is on the other end of the molecule. That's at the acid part of the fat. The acid part of the fat, which is this part right down here. This chemical group on the end is a carboxylic acid. It's a group, we've seen them before. They were the end of an amino acid. It's also the acid in an amino acid, is the carboxylic acid. It has a carbon that's attached to a carbonyl group and an acidic, a slightly acidic hydroxyl group, meaning acidic, meaning this hydrogen it will come off of here very readily. It'll ionize. That acid, the acid end of the molecule, is polar. It's hydrophilic. It likes to be in water. So just because some fatty acids can get really long, we don't draw out that whole molecule. You can abbreviate it like this. So you can see the abbreviated structure. You have CH3, which is the methyl group on the end. The end of the molecule always ends in a methyl group. And we see that down here, the CH3. And then there's a bunch of CH2 groups in the chain. Yep. Yeah. Mm-hmm. Yep. So there's a hydrocarbon end, a long, greasy tail, and then a small polar end. So you can imagine this molecule. None of this bit of the molecule down here wants to interact with this part up here. This is very polar. This is nonpolar, completely nonpolar. Because this part of the molecule does not want to interact with this part of the molecule, these kinds of molecules do interact with one another in a very ordered or regular way. Such that they like to line themselves up so that these long greasy tails line themselves up next to each other because they like to be around other hydrophobic molecules. And then these polar parts also like to cluster together. And when you do that, when you line a whole bunch of them up, you can get these layers, micelles, or in a biological context, a cell membrane. All the membranes of our cells are made up of a type of lipid that we will get to, phospholipids, glycerophospholipids, that we'll see the structure of later. But in general, we have these greasy tails, which are the sort of fingers that are sticking down. They all line up next to one another. And then the yellow and red balls on the top, those are meant to represent the polar head groups, the fatty acid parts that will line themselves up. And because of this, they can form little bubbles that are called micelles, or large ones that can be organized into cell membranes. All right. So next is the notion of saturated and unsaturated fatty acids. So a saturated fatty acid is a fatty acid that doesn't contain any alkenes. An alkane is a hydrocarbon where all of the bonds are single bonds. All right, so like this part down here of this molecule I'm holding, this is all alkane. And this would be a saturated fatty acid. Each of these carbons is saturated with hydrogen, meaning there's as much hydrogen on them, they've absorbed as much hydrogen as possible. If we're going to unsaturate a fatty acid, we're going to remove some hydrogen from it. And by doing that, we end up introducing a double bond. We incorporate a double bond between two carbons, and we'll take a hydrogen off of each of those carbons. So here's an example of an unsaturated, what would be called a monounsaturated fatty acid. I'm going to hand this around. Please don't take anything off of it. Thank you. But take a look at it. You can see how those carbons involved in the double bond have less hydrogens on them than all the other carbons, and which is why we call it an unsaturated fatty acid. So saturated and unsaturated fatty acids are found in nature naturally, and all of the unsaturated fatty acids that you find in nature are going to be called cis fatty acids, and I'll show you what that means in a moment. When you're drawing the line structure of them, you just put a double bond in there, like so, as you see. And you can you can draw them in this sort of abbreviated structure here, where this subscript here, 7, this just means that there are 7 CH2 groups in that chain, and on both sides, for example. All right. So you can have a monounsaturated fatty acid, which is like the one that I'm passing around, or you can have polyunsaturated. I didn't have enough double bonds with me in my bag today to make a polyunsaturated fatty acid, but it would simply have 2 double bonds or more. You can have more. So when you are categorizing unsaturated fatty acids, you categorize it using what's called the omega system. And when the way that works is the last carbon in the chain is called the omega carbon. Omega because omega is the last. Just throw it. Yeah, the last carbon in the chain is the omega carbon, and you just count the bonds up from the omega carbon. Thank you. That the double bond occurs at. So this fatty acid here would be an omega-6 fatty acid, which is actually omega-6 fatty acids are fairly common. So yeah, I'll come back to your question in a minute, Theo. Just hang on to that. Okay. So unsaturated fatty acids, the structure of this double bond, I'm going to hand these around again, and I want you to take a look at this. The structure of the double bond, you'll notice as you're inspecting them that all of these bonds are pretty flip-floppy. They can rotate around, right? I can rotate these bonds around. The only one that I cannot rotate the orientation of is this double bond. I can't spin around this bond. It's fixed, right? So because of that, the double bond itself can be categorized either as a cis or a trans double bond. Cis is one in which the 2 chains are on the same side of the double bond. So if you imagine the double bond is like a plane. If the double bond is a plane, both of these are on the same side of that plane. All right, this is a cis double bond. These are the ones that occur in nature. And you may think to yourself, well, that's kind of stupid. Why that? Because it puts this weird kink in the fatty acid that would be sort of more convenient and easier to pack that fat in if you didn't have this kink in the middle of it. So I'm going to hand this kinky fat around to you. There you go. And if you compare it to the trans, so again, looking at this plane, looking down the plane of the double bond, the chains that extend away from it are trans, meaning they're on opposite sides of the double bond. They're on opposite sides of that plane from one another. Trans fatty acids are, turns out, they're lower in energy than cis fatty acids. And if you take a cis fatty acid and heat it up, you can actually drive a cis fat to a trans fat. You can give it enough energy to overcome that barrier to rotation and become a trans fat. So a lot of the cis fats that are in your diet are really good for you, right? They're like fish oil and the oils that you get from various vegetables like olive oil or peas or kale or all these foods that are healthy for you. But if you heat them to very high temperatures, you can convert cis fatty acids into trans fatty acids. And trans fatty acids, I'll talk about them in a little bit, but they're associated with heart disease and a lot of the health problems that go along with deep fat fried foods, right? And this is where a lot of trans fats come from. So as you're taking those around, you can sort of see the difference between a cis and a trans fat. Does everybody sort of understand the molecular origin of this concept? It's not super difficult. Yeah.
There's still like a lot of trans fats.
Oh man. Yes, absolutely. Yeah, so in the last chapter, I guess I shied away from driving the point home about the prevalence of readily available sugar in the American diet as divorced from fiber. I did say it, but I didn't. I have a whole set of slides that I decided to not show. But one of the reasons that America is in this crisis of metabolic disorder, there's been a radical increase in obesity in America in the last 40 years. And that is essentially because America has developed the ability to present all this fructose, really, but sugar in general, into the diet in the absence of fiber. And that's one of the things that is behind the metabolic disorder and heart disease and obesity, et cetera, et cetera. The other part of that, however, is the prevalence of trans fats in the American diet. Now that has gone down. It has gone down in recent years. People are becoming a little bit more aware of it, but it's still out there. Absolutely, it's still out there. So every time you eat some French fries, you're eating some trans fats. Every time you have some potato chips, or anytime you have some, pepperoni pizza or donut that's been deep fried, or on and on and on. Any kind of deep fat fried food is going to have some amount of trans fats in it. It's hard to avoid. Yes, sir.
What about just a stir?
Yeah, stir fry is if you're just using. So when you're making a stir fry, hopefully you're using an oil that has a high smoke point, so that you're minimizing the breakdown of the fat itself. And as you do that, the stir fry is not reaching the same temperatures that a deep fryer is reaching, because as you're stir frying, even if you are going at a high flame and a pretty high temperature, it's still it's being cooked at the temperature of the food that's in there has all this water in it, and so that water is dominating the temperature profile of the stir fry. Whereas in a deep fat fryer, you have such a huge thermal mass of really hot oil that you're reaching much higher temperatures and a much higher concentration of fat than you would in a stir fry. Stir fry is actually a fine way to cook because there's not the same intensity of fat in the preparation. And then secondly, there's much more water, which is going to, that water, no matter what, if you have water in a pan that's cooking, even if it's boiling off, it's still at the temperature of boiling water, right? You can't get water above the temperature of boiling. So deep butter does not have to be a trans fat, but it's really easy to get trans fats out of butter. So for example, the technique that you often will see where people brown butter. Has anyone ever had browned butter? That is, you're certainly creating some trans fats when you do that a little bit. You're also, the browning is actually the Maillard reaction. However, in the fat, you're dry. It's the same. Yeah, the way to brown butter is to do it, to brown it, but do it before you get the butter to the smoke point. If you're getting the butter to the smoke point, that's when you're getting trans fats. It's the browning of the butter is the Maillard reaction, which is the reaction between the proteins and the carbohydrates that are in the butter. Yeah, and that gives it that nutty flavor that people like so much. but it's a tricky thing to do. A lot of times people go too high in temperature and brown it too quickly, and if you're starting to get smoke off of it, then you're getting trans fats in it. Yeah, there's going to be some trans fats in butter anyways.
Is there a reason?
Trans fats don't necessarily taste better. It's one of the aspects of taste, like the two heat reactions that lead to flavor profiles more than anything are the Maillard reaction, which is the reaction between carbohydrates and proteins, and the caramelization reaction, which is the reaction between carbohydrates themselves. And it just happens to be that you can get to the temperature that drives those reactions very well in the presence of fat, because you can get a liquid fat to a higher temperature than you can get liquid water, right? You can't drive the Maillard reaction and the caramelization reaction at the temperature of boiling water. It needs to be higher. So in the presence of fat, it's one way of doing that. So they've been talking about, I don't know if you guys follow food trends, but have you heard about the air fryer? These air fryers meant to be ways of getting food up to that temperature to drive the favorable reactions without incurring the problems that you have with trans fats in deep fat frying. I haven't played with one of them. I don't have one of them, but I think it's pretty interesting. I've also heard that it does not work as well for all kinds of foods, compared to deep fat frying. But yeah, the thing that tastes good about these trans fats is, the trans fats themselves. I cut this lab out. Fats themselves do have smells and a certain flavor profile. So a trans fat is going to have a different smell than a cis fat, perhaps. And maybe that's evolutionarily related to the benefit of cooking meat. When you cook meat, you're eliminating pathogens, and maybe we evolved, humans, when we were developing that, sense. That was before we were living to be 70 years old and having to worry about heart disease. I don't know.
When will we live to 120?
I don't know. I'm not.
I thought you had the answer.
I don't. 120. It could happen. It could happen in your lifetime, man. Telomerase, when they figure out exogenous telomerase. Okay, keep on with the lipids here. Alex, let me give you a chance to contemplate some trans fats too. Okay. So here is just a list of some common fatty acids. I'm going to go through this list in subsequent slides, but I'm just showing them all in the same slide here so you can get a sense of comparison. So there's a number of different ways that these fatty acids compare to one another. So as you look at this list, this is not an exhaustive list of all the possibilities, the structural possibilities that there are for the fatty acids. These are just the ones that commonly occur in nature. Okay, and so one of the things, the characteristics that you'll see are chain length, number of double bonds, and exactly where that first double bond is located. And so as you look at that, one of the things you'll notice is that the chain length is always in increments of 2. They're always even numbers, right? They vary by numbers of 2. And that's not a mistake. It's because biology, the way biology has learned to utilize fats, is by clipping them down 2 carbons at a time. So if I were to pull out this fat here, let's just get it in a nice. Trans fats really are easier to work with, aren't they? Let's just get it into a reasonable structure here. As we clip, biologically, if I was going to be eating some of this in a cell, my cell is going to utilize this. I'm just going to chop this part off right here, right? And that would be a hydrogen sticking out. And that essentially looks the same as the fat before, right? I've just taken this chunk off. It's just shorter now by 2 carbons. Without going through the detailed metabolic pathway as to why that actually is molecularly, just take it for granted that it is. So chain length. And chain length is probably one of the most, the strongest determinants of the flavor profile and smell of the fat. So the shorter a fat is, the shorter that chain length is, the more unpleasant that odor is to humans. And that is because these longer chain lengths, which are found in a lot of meats, as you shorten them, that is an example of rancidity. So say you have eicosapentaenoic acid, EPA, that the second from the bottom there. That one is in fish. A lot of fish have that. And when fish is first taken out of the water, it smells fresh. As it sits there, those fats rapidly get broken down by the microbiota that are on the fish. As it cleaves chunks off of that chain, those chains, the EPA is getting broken down into shorter and shorter fatty acids. That's what smells nasty to us. So like caprylic acid, for example, is kind of odoriferous. Also, you'll see the number of double bonds, whether it's it's called the degree of saturation. So oleic acid has 1 degree of unsaturation, whereas linolenic acid has 2. Something like DHA, docosahexaenoic acid, has 6. Exactly where those double bonds are located in the chain is also an important characteristic of fats. So let's walk through that first, that list a little bit, and look. The first thing that separates fatty acids is the saturation versus unsaturation. So these fats here, C12, 14, 16, and 18, these are the common saturated fats that you're gonna find. So this includes all animal fats, are saturated fats. Human breast milk, coconut has a lot of saturated fat. Palm has a lot of saturated fat in it. These fatty acids tend to have higher cook temperatures. We'll get into why that is in a bit. All right. So then all other fats are unsaturated, and they can either be mono or poly. Here are some examples of the mono-unsaturated fatty acid. So an example, the most common example is oleic acid. And we find this, it's called oleic acid because it's used to make oleo, which is sort of like the Italian precursor of what we think of as mayonnaise. But it's in olive oil. So anywhere from 55 to 83% of olive oil can be oleic acid. It depends on where the olive oil was grown, what strain of olive it was made from, how it was processed, etc. As an aside, most olive oil that you buy that's imported from Europe, commercially, like at Hannaford's or whatever, most of it is actually not olive oil. Most of it is, it's usually a blend of a little bit of olive oil, and they put a lot of safflower oil in there to sort of water it down and make it more inexpensive. If you are looking for pure olive oil, the stuff that's made domestically in the United States, you're more likely to find an olive oil that's pure olive oil. Pecans are also really high in oleic acid. I don't know if anyone eats a lot of pecans. They're kind of expensive, but really good for you. Canola, peanut, macadamia, sunflower, grape, poppy seed. Okay. There are some vegetable sources or the animal sources of oleic acid. So in poultry and a not insignificant amount of oleic acid found in lard. All right, that's that. We'll come back on Wednesday. I'll talk about polyunsaturated fats. We'll talk about trans fats and then move into triglycerides. Questions?